Thank you.
All right. Thanks, Alana. Can you hear me okay?
I can hear you. Thanks for being here.
Okay, great. All right. Hi, everyone.
Hi.
Thanks for taking time. And if you've got questions along the way, I'm also fine with people just piping in, giving those questions here. All right. So this is our work for the Happy Lungs about novel structure-based assessment of genomic mechanisms of RET TKI sensitivity and resistance. And I'd like to particularly thank the people in my lab, especially Monique, who's helping lead this. All right, so I'll go through the basic clinical framework pretty quickly. People are familiar now. Right now, our first line therapy is selpercatinib and pralsetinib. There are new ones in the pipeline. People are familiar here with the response rates. Both drugs have high response rates here and are effective drugs, but we do know that commonly resistance does eventually emerge. Now, I think everyone here knows that there's a lot of different RET fusion partners. So we're showing here, so this is from a large analysis that's been put together of all the different RET partners. And KIF5B is the most common, as we'll show later, within KIF5B RET fusions, there's a whole bunch of different breakpoints. CCDC6 is the second most common. And then you see the list going down there, which includes some really exceedingly rare ones. I think there's data supporting that there may be differences in the fusion partners, but this is obviously a question that we're interested in investigating and whether the specific fusion partner should guide therapy. So our goal here is to be able to match the right inhibitor to the right patient. And so to investigate how unique RET fusions can impact the sensitivity. All right, and just to talk about approaches we've taken before for EGFR. The question is how we should classify it, and I think everyone's familiar with the fact that if you've got a lot of different items, you can classify these, alphabetically, like here, ant, airplane, and alpaca together, beetle, bus, and Bugatti Veyron. You can do them by frequency, so just like we talked about KIF5B RET being the most common and then listing the least common ones down there as well, or by structure function. And I think if our goal is to find the right inhibitor, this is the direction we would like to head to, where we're able to, regardless of how common or uncommon a fusion is, to be able to match it with the right drug and use this also to understand resistance mutations. So really develop a common structural understanding of the fusions and the way resistance emerges in a way that helps us select the right therapy, and then hopefully develop better therapies once we understand the limitations of our current ones. So our group has, and in addition to Monique Chamang in our group, has been leading a lot of this analysis. So we've set upon making, I think, what's the largest set of different models of all the different breakpoints and fusion partners. So we've constructed these 19 different fusions, and you can see a lot of them are KIF5Bs at the common fusions, the 1512, 1612, and so forth. And then the most common fusion partner with all the others. In some cases, some of these rare ones have only been seen once or twice. We're also using RET fusion positive cell lines, including the MDA-L30, the LC2, which is a common cell line that's widely used, and the NCI-TH1101 cell line. Those last two are both KIF5B RET fusions with the relatively common 1512 fusion breakpoint. Okay, so the first thing, and I think this is reassuring, is that when we look at the BAF3 models and we look at the patient-derived models, the MDA-L30 or the here, we see generally a good concordance in the drug sensitivities between these. So in this particular case, you can see the most potent drugs include pralsetinib, ponatinib, and sulpercatinib. Less potent drugs include vandetanib and cabozantinib, and that matches up pretty well between them. Now, of course, we know a patient-derived model has the advantages that it's a human model. It includes other co-occurring genomic alterations that can vary between people, whereas the BAF3 model is a pretty a clean model with relatively fewer genomic alterations, but it's in a mouse background. You know, I'll also mention that we've developed a human model that we're looking at using to a greater extent with a classic EGFR mutation, where we've knocked out the EGFR mutation. We've made them dependent on IL-3 receptor and IL-3 to keep them alive, like BAF3s are. And then we can put in other genomic drivers. So essentially, it's taking a human cell line and making it like the BAF3 model. So that's another model that's potentially available. Oh, sorry, my system just crashed. Okay, so the first point now is, with our look at all the different breakpoints and fusion partners, we do see that there are differences in the sensitivity to different RET inhibitors. So here, if you look at the KIF5B RET fusions, so green, blue, purple, most of those are more sensitive. The 1612 is less sensitive, and we see that reproducibly. But when we look at sulpercatinib does not work well for the CCD6 fusion. This is the C1R12 fusion. But we don't, that's for sulpercatinib, but we don't see that difference for pralsetinib. So this suggests that if you've got a CCD6 fusion, that pralsetinib may be the preferred drug in that particular case. And when we look at cabozantinib, we once again see a different pattern of sensitivity, although in the case of cabozantinib, it's less potent across the board for all these different fusions. And again, these are all now primary alterations, not resistance mutations that come up later. So we've put them here in this heat map. And so now we've taken all the common breakpoint fusions. And again, this covers well over 95% or so of all the fusions that have ever been reported. And we've screened it against the drug sensitivities for sulpercatinib, pralsetinib, and all the other drugs that have been reported clinically. Now, in this case, because sulpercatinib and pralsetinib are more potent, that sort of washes out the color here, but you can normalize it in a way that you can see the differences between sulpercatinib and pralsetinib. They work well for most. CCDC6 is the one that is separated between the two. That's less sensitive to sulpercatinib, much more sensitive to pralsetinib. But for these other drugs that are available, you see different patterns of sensitivity and resistance. And I'll bring to your attention zetalutinib here that is also generally potent across the board for the vast majority. Interestingly, for the CCDC6 fusion, we see that it promotes resistance to a lot of the different drugs, including cabozantinib, regorafenib, vandetanib, but there are a couple drugs that are quite potent for it, like the TG101, 348. So I think this gives us a good starting point for considering that we may be able to tailor at least some of the drugs based on the fusion. But fortunately, for the starting point with our two potent inhibitors, CCDC6 is the main one to take into account. Okay, now moving on to mechanisms of resistance for the RET inhibitors, and thinking about which drugs remain effective in resistant cells. And just to remind everybody, we have RET-dependent resistance and RET-independent resistance. And we've got different models that are appropriate for both of these. In RET-independent resistance, we're focusing on signal bypass pathways. Obviously, for RET-dependent resistance, we're looking at other RET alterations. And so here I'm going to introduce the lenti-mutate system. And this is really work driven by our great collaborator, Rolf Kittler. And I'm not sure if is Rolf on the call with us here?
Yes.
Okay, great. So I'll introduce this and I'll let Ralph give a bit more detail about this. But the way I would liken this, we historically have done our cell line selection work by taking a given cell line, incubating it in a drug for a long time, and then occasionally one or another alteration will arise, promote resistance. But we know that that is not a great way to investigate all the different resistance mutations that can happen, because a given model will often have a propensity to go one way or another for resistance. So, for example, those of us that have worked for a long time in the EGFR space know HCC827s will commonly go one way for resistance, PC9s will go another, 406s will go another. So that means that our ability to scan the full space of resistance alterations is limited by the models we have and the propensities that those particular models have, which may be dictated in part by co-mutations or things like APOBEC expression levels and so forth. So in the same way that if we want to, investigate different ways that adaptations can occur, you could pick a particular niche. And here I think about the Galapagos, where all these different finch beaks evolved, and they evolved to fit different ecological niches in the Galapagos Islands in terms of, the ability to pull seeds out of little rocks or out of bark in trees and so forth. So you could spend time looking at all the different niches in the Galapagos if you want to understand all the different ways that they could evolve. But imagine if you could just take all the different possible beaks, that could arise in the Galapagos finches and then release them all at once and let them compete. So instead of taking hundreds of thousands of years, to test what the best adaptation is for a given environment, you may be able to figure that out in a couple of generations. So that's sort of the way I think about the lenti-mutate system here. Here, Rolf, do you want to briefly describe some of the advantages here of the lenti-mutate system?
Yeah, so the biggest advantage above other mutagenesis approaches is that we eliminate the need of making libraries. Yeah, so when you think about now, how could you generate, let's say, all possible substitutions in an open reading frame? So there are different ways to do that. Yeah, you could just basically synthesize them, and people are doing that. You can use very fancy approaches like base or prime editing. Yeah, but for that, basically, you would need basically relatively large libraries of targeting vectors, or as I said, of mutants. So what we actually found is that we can actually tweak lentiviral transgenesis to introduce basically a large spectrum, if not all possible substitutions and other mutations during transduction. And that basically means, so we package, so basically in the case of KIT5B RET, the ORF into a lentivirus, then we transduce basically the cells with that, and we use then basically a mutated reverse transcriptase that has a relatively high error rate. And then during transduction, when the RNA is reverse transcribed into cDNA prior to integration into the nucleus, basically a lot of substitutions are generated randomly, and then from the resulting population of cells, we can now select those mutants that confer resistance. Yeah, and we can relatively easily scale that up. So let's say if we transduce 10 million cells, then based on the mutation rate, we could guesstimate that we would basically generate all possible single nucleotide substitutions in the open reading frame. So then we select that now with RET targeting drugs such as pralsetinib or selpercatinib. Then we basically recover the surviving cells that now carry basically the resistant alleles, and these we can then very easily recover just by performing next generation sequencing and basically for this open reading frame. So we don't need a very large number of reads. The sequencing is actually very, very cheap for that purpose. And that's overall a very facile and very cheap approach to detect on-target resistance mutations.
Okay, thanks, Ralph. And so the key advantage is you're screening literally millions of different mutations at once and then seeing which ones can survive and grow. So, so you can see that the information that took us many years of clinical trials, led from people like Vivek Sabaya here at MD Anderson and Alex Drillin on the call here as well. In the period of an experiment that takes a few weeks, or a month of constant work, we can identify all these same mutations. So here we can see that selpercatinib resistance can emerge, and here we're showing the frequency or the fold enrichment when you grow this mutated library with the drug. We can see we get the 806, 804, 810 mutations. These are ones that have been seen clinically. And this 918T mutation here as well for selpercatinib. Now, differently with pralsetinib, we see some overlap. We see, for example, the G810S mutations, and that's one that was seen with selpercatinib as well and had previously been described. We see the 806s here as well, but we see a couple of additional novel mutations, and we don't see the 918T. So this begins to give us insights into, which ways can resistance emerge, and does the same resistance mutation cause resistance to everything? Now, you may say, well, we don't know if those mutations occur clinically, because maybe clinically there are reasons that every possible mutation wouldn't occur. But we can tell you the ones that we're seeing are, so everything that we see clinically, we tend to get from the screens, but occasionally we do get additional things that have not been seen clinically. It remains to be determined if we just haven't collected large enough data sets yet, or whether there are reasons they may not occur. Now, as you're going to see, we then validate these mutations using independent systems here. So we've now taken all these resistance mutations that have been identified from the lentimutate screens, and now we've engineered them into the KIF5B RET, and we're using, again, the common 1512 fusion. This is the fusion that we also see in the MDA L30 cell line as well. So that lets us, also transfect them in there here, if we like. And so here now we can then transfect them in, make stable cell lines with that particular fusion, and then look at the impact of the mutations on our ability to inhibit RET. So you can see here that the vast majority of these do, in fact, induce significant resistance. I'm sorry, here we're showing just the expression that we're seeing phospho-RET in all of them. We're activating downstream signaling, and then we're going to next move to sensitivity. Okay, so the 810 mutation, consistent with what had previously been shown, does induce resistance to selpercatinib and pralsetinib. So if you look here at the heat map, this gives you sort of the summary information. If you look, for example, at the G810R mutation, you see that's resistance for most of the mutations across the board. Now, there are a couple of drugs that can hit that mutation, zetalutinib and the ON12330. can inhibit that mutation. We're seeing a lesser degree of resistance with some of the other alterations, the G810C, for example, and this is all compared to the wild type, if you will, KIF5B RET fusion that doesn't have any secondary mutations. So you can see here, this information could then help guide that, okay, we can confirm that these mutations do cause resistance to selpercatinib and pralsetinib, but now we've got a couple of drugs that could potentially be used in those cases. All right, here we get into all the additional details now with these different mutations. So you can see if we put this on the KIF5B 1512 fusion backbone, and you look at the top on the heat map, that tells you what the regular 1512 fusion looks like without any secondary mutations. And then you've got selpercatinib and pralsetinib. You can see virtually all of these mutations induce resistance to one drug or the other. Now, the one that doesn't validate here is the M918T. Now, interestingly, there's some independent data that 918T may just help cells grow more aggressively, but doesn't necessarily confer resistance. So in other words, what may be happening is we may be getting an increase in oncogenicity, but it may still be a sensitive mutation. So that's something we're validating right now.
And John, if I may add that,
Yep.
So one basically further refinement, so we are thinking about for lentimutate, it's also possible that in some cases we introduce two mutations, so the M918T might have occurred actually with some of the other mutations, yeah? Currently, the way we're doing the sequencing, we can actually say that. Yeah, that is actually a possibility. We had M918T on the background of some of the other mutations, and then the cells might have grown faster and been resisted. Yeah.
Yeah, thanks, Ralph. So what we've got here now, I think for the first time, is we've got the most comprehensive look at resistance mutations that have been reported. We're able to validate the vast majority of these, that they cause resistance. And for the first time, we can start distinguishing between selpercatinib and pralsetinib in terms of which resistance mutations arise in each and which could be used to treat the other. So for example, if somebody were to get the 804M mutation when they were treated with selpercatinib, they could easily switch to pralsetinib. If somebody were to get the 804E, the 730I on pralsetinib, they could easily switch to selpercatinib. But some of these mutations, like the 806N or the 810S, would require having to switch to an entirely different type of drug altogether. All right. And so these are grouped here into the categories of selpercatinib and pralsetinib resistance together, selpercatinib resistance alone, or pralsetinib resistance alone. And then you can see across the board which drugs are effective in these different alterations that arise. You know, and you can see we do have some different options, and zedalenib is one that looks particularly favorable here. All right, let me go through now RET independent resistance mechanisms. So here we're looking at cellular adaptations, and you know, we've learned a lot from other models, you know, EGFR and ALK and so forth. One of the things that often happens is when you inhibit a tyrosine kinase, you get a compensatory upregulation of cell surface signaling from other RTKs that are there. And that sometimes doesn't require that they're mutated. So for example, what's been shown in other systems like KRAS, if you inhibit a KRAS G12C mutation, you can get compensatory signaling through wild type EGFR. So we've put a look here, especially on wild type pathways that you wouldn't detect by just looking at genomic mutations here. All right, so here, and I'm gonna not present all the data, but there's been suggestions from work from other groups as well that EGF promotes RET inhibitor resistance through wild type EGFR signaling. And we do see some evidence of VEGF receptor expression as well. Now, I'll also point out that selpercatinib does have some activity against VEGF receptor 2, at least. So here you can see that if we take it as compared to wild type pralsetinib, the addition of EGF ligand does promote resistance here. VEGF itself doesn't. And this is to both of them. Now, again, this is through wild type EGFR signaling. All right. And so here now in the LC2 model with the CCDC6 RET fusion, you can see with the addition of the ligand for EGF, we can block that both with erlotinib and poziotinib. Now, why do we choose these drugs? Well, you may say, well, let's pick one that's more specific and less toxic. But the reason they're less toxic is because they don't hit wild type EGFR. And here, wild type EGFR is exactly what we need to hit. So that's why we've chosen drugs that do have that activity against wild type EGFR. So you can see here combinations of erlotinib and poziotinib can enhance the efficacy. Erlotinib and poziotinib by themselves, as you'd expect, are not active because we still have RET fusion signaling that's occurring. So let me just try to make some broad points here. Unique RET breakpoints and fusions have distinct sensitivities. And here we're just highlighting the CCDC6 with selpercatinib as a particular example. So we can't have a one-size-fits-all for RET fusions. The BAF3 RET fusion models can predict tumor cell sensitivity, and we now have dozens and dozens of these models. About 20 of them are the baseline fusions, but now we've made lots and lots of the potential resistance mutations, and this should really facilitate the study of any new drugs that come along for RET inhibitor resistance. RET inhibitor resistance can be mediated by secondary RET mutations, and we believe we've now comprehensively characterized to a deeper extent than ever done before selpercatinib and pralsetinib resistance, at least via RET. These resistance mutations are also drug-specific, so not every mutation confers resistance to every drug. And then we've identified some drug combinations. I didn't present here, but we also have data about mTOR combinations that can improve the efficacy of RET inhibitor combinations. Okay, so with that, I apologize. I know I had to go fast and cover a lot of data, but I'm happy to stop and take questions here.
Thanks so much, Dr. Haymack. So just a reminder that if anyone has like any question, can type it in the chat. And if there's not time for questions, we can always like get in contact with investigators and get the answers back to you by email as soon as possible. So, well, for the sake of time, maybe we can move to the next speaker, Dr. Trillon. And I don't know, Dr. Trillon, if you can share your slides. Let me know if you have. Yeah, thank you so much.
Set this up. Can you hear me well?
Yes.
All right, great. So thanks, John, for that great talk. I think it really set the stage for a lot of what we're going to discuss in this next section for the other module or project that we have that was very graciously supported by the Happy Lungs project. And as you'll see, the goal of this project was really to study targeted therapy resistance in patients' cancers that are dependent on RET. So this is a lot of data from patients treated both on commercial supply with these selective RET inhibitors or on clinical trials. Okay, this is another way of showing you the data that you've seen from Dr. Haymack earlier. And basically, this is a plot showing how well these approved selective RET inhibitors can work, both in patients that have not had any treatment for their advanced RET fusion positive lung cancer, or perhaps patients who have had chemotherapy, such as a platinum doublet, prior to getting the targeted therapy. And what you need to know is basically that if you fall into the right upper quadrant of this graph, that means you're seeing a very high likelihood of knocking the cancer back and shrinking it, and also a high likelihood of, in addition to doing that, keeping the cancer under control for a very long time. And those metrics, the technical terms are response rate and progression-free survival. So here you see that both selpercatinib and pralsetinib on the programs that got these drugs approved have very good activity, which I'm sure many of you know. And this next slide really highlights that these benefits extend beyond lung cancers that are dependent on RET. And this shows the waterfall plots, which are simply plots of individual patients and how much their cancer is shrinking with a therapy. So on the left, you have the lung cancer patients, and we talked about how selpercatinib and pralsetinib work very well for those cancers. But you also know that these RET fusions or mutations mainly are also found, in thyroid cancers. So you see RET fusions in papillary thyroid cancer, you see RET mutations in medullary thyroid cancers. And there are many other cancers that have RET fusions, like gastrointestinal tumors, colorectal, pancreatic cancers. And fortunately, all of these patients are now encompassed by the FDA approvals for selpercatinib in particular. So you have the approvals of both drugs for lung and thyroid. And then just last year, we have what we call a tumor agnostic approval for selpercatinib for any cancer that harbors a RET fusion. But for balance, it's important to point out that we've observed with pralsetinib also across different cancers that we're seeing the same effect, that it can work very well. So what are the goals of this project? So like I said, the major goal is to look at cancers from patients who are treated with targeted therapy or other therapies. These are cancers that have either RET fusions or mutations. And while most of them are lung cancers, we are also looking at thyroid cancers and other tumors. And the first major aim is to really get a good idea in these clinical or patient samples of what we're seeing in terms of resistance. And all of this was discussed by Dr. Haymack in the last talk. And the second aim is to move away from targeted therapy and to look at how well our other treatments work in RET-dependent cancer. So this first section of the talk will discuss the first aim. And how did we do this? Well, thanks to the support from Happy Lungs, many people from different countries got together to form a registry with a very nice name, the RETgistry. And these are essentially aggregates of data from patients with either RET fusions in their cancer or RET mutations in their cancer. They should have gotten targeted therapy or another therapy for advanced disease. And what we did was we looked at the gene sequencing of tumor or liquid biopsy samples that were sent from these patients to see what patterns of resistance we could find. Now, this is just walking you through the process of how we got the registry together. Thanks to Jesslyn, who from Mass General, who couldn't be on this call, but she had spearheaded setting a lot of this up with a REDCap database where everyone would enter their data. There was an IRB-approved protocol, very careful identification of each of the sites or hospitals with the appropriate documents and protocols that allow the use and sharing of the data. And then finally, all of this data went into that singular database and was subsequently analyzed. So this is to give you a little bit more of a granular look at how many hospitals have agreed to participate in the Happy Lungs project. And you'll see here that we're very fortunate to have many investigators from different hospitals around the world. You'll see here representation, of course, from the United States, but also from Europe and from Asia. Many of these researchers are lung cancer researchers that have contributed a lot to the field and have the clinical trials, and so have the patient base to contribute cases to this registry. And this data was already presented. So this was presented at the annual American Society of Clinical Oncology meeting by Alyssa Cooper, who's from Mass General and works with Dr. Lin. So this is as per a prior data cut, where we had more than 100 biopsies, either tissue or liquid biopsies, in almost 90 patients who were treated with either selpercatinib, pralsetinib, or both drugs in sequence. And I should point out that while we're talking about this data set, since then we expect perhaps another 100 cases to join the database, which are currently being entered in the system. So going back to that data cut where we had almost 90 patients, you'll see here the demographics and features of these cancers. So expectedly, most of them were lung cancers. And the second most common group in terms of cancer type was thyroid cancer. You'll see the different fusions from these patients. You've heard these from Dr. Haymack previously, KIF5B RET, CCDC6 RET, and for the thyroid cancers, the MTCs or the medullary thyroid cancers, of course, those harbor these kinase domain mutations like M918T. You'll see that some of these patients got targeted therapy as their first line of treatment, others got it as second line or third or greater. And we looked at a variety of different factors beyond just the RET alterations, which we'll discuss in the subsequent slides. So you've all also heard this from Dr. Haymack. This is how cancers can develop resistance to targeted therapy with selpercatinib or pralsetinib. And one of the very first published instances of this is on the left. This is a paper that we put out spearheaded by Dr. Solomon from Australia, where that tricky mutation, the RET G810 substitution that Dr. Haymack mentioned and studied, was found in a patient who had gotten selpercatinib, and it was found in many areas of the cancer that were sampled. Now, just to break this down again for you guys, these mutations can accomplish a number of things. When I try to explain this to patients or their families, I say that what happens is the mutation could either put up a blockage that prevents the drug from binding RET, but it can also do other things. So it can change how hungry the protein is for ATP, which sort of feeds its growth. And based on what we know from other fusions, there may be other mutations can even change what we call the conformation of the RET protein. So it looks different and has a different topology. And so you may need a different drug altogether to bind it. And this was mentioned by Ralph earlier, that there are some mutations that can be acquired in sequence. And we've seen this most with tumors that perhaps acquired one mutation first or had a mutation to begin with. Let's say if they were thyroid cancer, were then exposed to targeted therapy and then developed another mutation on top of that. So here we have an example of a thyroid cancer that first had an M918T mutation and then developed the gatekeeper 804 substitutions, both of which were on the same strand of DNA, thus really impeding the binding of selpercatinib. So going to the registry, I'll have you focus here on the red slices of the pie. And the top line result here is that we do see these mutations in patients who develop resistance to targeted therapy, but you don't see them with a very high frequency. They're not super common. So in this data set, you'll see that these RET-dependent resistance mechanisms occurred in about 13% of cases. There were prior reports from Dr. Gaynor and from us where we showed that the likelihood of acquiring these mutations was less than 10%. In the pie chart on the right, you'll see the distribution of the different mutations. Most of them, so more than half, were that really tricky solvent front mutation that Dr. Haymack mentioned. And it's still encouraging that we're finding these mutations. but it highlights that we have to look very carefully to make sure that we don't find anything else in the cancer, because the newer drugs that Dr. Haymack spoke about that were really designed to target these mutations were more specifically designed to target resistance of that tricky solvent front mutation, for example, and maybe some other mutations like the gatekeeper. And while these are encouraging, I think our early experience is that you really need to match someone's cancer very thoughtfully to these new drugs. So it's important to resequence the tumor, make sure that you don't have bypass resistance or resistance that's not dependent on RET solely anymore. But in cases where perhaps we don't find a smoking gun or a culprit for resistance, or you find one of these mutations, we found that these patients perhaps stand to benefit more than those that acquire the other type of resistance that's independent of RET. So we'll see what happens with these next generation drugs. Some of them actually have already, they started early in clinical trials, but have since withdrawn testing based on early results. So we'll see how this shakes out. But my hope is that one or more of these eventually get approved in patients who have RET-dependent cancers to offer more treatments across the board. Now, moving on to that other mechanism of resistance. We talked about cancers that are resistant but still rely on RET. However, there seem to be many more cancers that develop resistance where they may be reliant on RET, but they kind of phone a friend and activate another protein made by a different gene. And in that case, it's a little bit more challenging to develop new therapies. So you see here, it's a very busy slide, but I'll just summarize and say that there are these other genes in the lower group of rows here, like MET, for example, or HER2, or FGFR, and even some other fusions that are not RET that have been observed to emerge as resistance mechanisms to RET-targeted therapy. So very interesting that we're seeing a wide variety of these across a number of different proteins. But there are some sort of convergent observations. So one of them, and this doesn't happen in all cases, is that in RET, and we've observed this with other fusions as well, there seems to be a preference for reactivating one of the major pathways that causes cancer by these RET fusions. And this is what we call the MAP kinase pathway. So here you see activation of other receptor tyrosine kinases that we talked about, RAS, RAF, and other mediators of this pathway that essentially turn this pathway back on and make the cancer grow and spread again. I said that there is the possibility of developing targeted therapy for some of these. And this is work that we did with Dr. Somwar and Dr. Ladani, who are on the call, and also with Dr. Yanni from Dana-Farber. But I mentioned the gene MET because we have found that there are many cancers with RET fusions that develop resistance to targeted therapy by acquiring more copies of MET or MET amplification. And it's a very busy slide, but we said, well, what happens if you add a MET inhibitor to the RET inhibitor, since presumably the cancer is dependent on both RET and MET? And that's what we did here in a variety of different models. So we looked at an organoid in the lower left with a combination of saprocatinib and crizotinib. We looked at this in other models on the upper left, run by Dr. Somwar. And maybe it's easiest to see the benefits of combination therapy in this plot on the right, where relative to the drugs given by themselves, either a RET inhibitor alone or a MET inhibitor alone, where again in these waterfall plots, you don't see shrinkage of the cancer, but you see growth. Conversely, if you give the combination in the pink and the blue bars, the cancer can start to shrink. And the obvious question is, does this work in patients? The answer is yes, and there are several cases of these already that have been reported. This is one of them that we had previously published in a journal. And this was a patient who had a RET fusion in their lung cancer, developed MET amplification, and then we added a MET inhibitor to the RET inhibitor, and the patient's cancer shrank. I think this is, in terms of clinical trials, a tougher space to operate, and a little bit more of an unmet need. We certainly have a phase 2 trial of an inhibitor of both RET and MET. You heard of cabozantinib, that's the drug from Dr. Haymack. It's not as great a RET inhibitor as the other drugs, selpercatinib and pralsetinib, but it's certainly something to consider if you have kind of that perfect situation where you rely on both proteins that are inhibited by this drug. But the hope is that we see more of these combination clinical trials to really expand the reach of targeted therapy for patients with this form of resistance. So to end with some of the data again from the registry, you'll see here that the two major forms of resistance, so reliance on RET and loss of reliance solely on RET in blue and red respectively. When we looked at the data in terms of when these occurred relative to how long patients were on the targeted therapy, there didn't seem to be a big difference. So this might change as we have patients who come off a targeted therapy after having been on it for much longer than those who maybe have been on for a shorter period of time, in which case there might be a difference between the types of resistance that emerge in the two cases. But stay tuned, especially as we're adding more patients to the registry. Now, this is work that Jesslyn and Justin Gaynor are doing. We're also doing at our center. If you remember that very first case of the solvent front mutation that was developed in Australia, the different sites that were sampled were from a patient who very generously, whose family agreed to an autopsy. And there are other cases like that where we can look at multiple spots or sites of cancer within the body and see if there are differences if you look from area to area at what the resistance mechanisms look like, what the genes look like on sequencing. So this is in process. This is a sample, an autopsy case from Dr. Lin, where you see, maybe just focus your attention on the right, the multiple areas that are sent for a very comprehensive sequencing assays, so whole exome sequencing, looking at DNA, and then RNA-seq, where they're looking at RNA. So we'll see what comes of that. We've also done a case here at Memorial where we're doing the same thing to look at heterogeneity, and hopefully we learn from these cases. So almost done with my slides. We'll very quickly go through the second aim, which was initiated after the first aim, so we have much less data. And to remind you, that's to determine how well other therapies, apart from targeted therapy, work in RET-dependent cancers, and specifically how well immune therapy works, because we've all heard about the benefits of immune therapy in lung cancer, but it seems like in cancers that have RET fusions, similar to some other cancers that have drivers in them, the benefits of giving immune therapy by itself aren't that great. So this was one of the first series that we published that showed that these cancers have a lower number of total mutations, which has been associated in part with the potential response to immune therapy. And maybe the more popular data set with many more patients, even though the exact numbers are actually smaller, was the ImmunoTarget Global Registry that we participated in on the right, where you see that there were very few patients that had the light blue bars that represent a response to immune therapy, immune checkpoint inhibitor therapy given by itself. But the field has very quickly changed. And we do have the approvals, as you know, of chemo and immune therapy together. And fortunately, this large trial, Libretto 431, has already read out. in patients with RET fusion containing lung cancers that received either targeted therapy or chemotherapy plus minus immunotherapy. And the first piece of information here was that perhaps unsurprisingly, the targeted therapy was the better therapy to give. Because if you see in the curves, targeted therapy in blue and chemo plus minus immune therapy in gray, you do see an improvement in how long patients' cancers stay under control with targeted therapy. But it's of interest to see what the difference is in the patients that got chemotherapy alone versus those that got chemotherapy and immune therapy. And at least on the first pass, it didn't seem like there was a very big difference between those groups. So if you think on that and the prior slide, it perhaps implies that you're really not getting a whole lot of punch, even when you add immune therapy to chemotherapy in RET fusion positive lung cancer. So, that we are trying to establish beyond the Libretto 431 trial, more real world data, looking at patients that have gotten both immune therapy by itself and immune therapy with chemotherapy. And one thing that we're going to try to do, if Lily and Loxo allow us, is to perhaps look at both the prospectively developed data set you saw for the phase 3 trial in tandem with this real world evidence that we're generating to see if there are similarities. And another thing we'd like to look at is, if there are more novel ways of turning on the immune system, novel immune therapies. It would be interesting if there were some patients in our data set that had very robust responses, that we look to see what those new strategies are to turn on the immune system and hopefully do more of that. So an update on this is that we are analyzing the data that we have on immune therapy with or without chemotherapy. But just this week, we were approached by Gustave Roussy. They're a very well-known cancer center in France. And they're also interested in this question. And we are in discussions to partner with them to add their RET registry to ours to look at this question of the effect of immune therapy. So stay tuned. But with that, I end my presentation and turn it over to our next speaker.
Thank you so much, Dr. Trillo. That was awesome. And the next speaker is Dr. Ruben. Dr. Ruben, if you can share your slides. Thank you so much.
Thank you, Renee. So hopefully everyone can see this properly. I appreciate the support from Happy Lungs, and I present this on behalf of Justin Gaynor, who co-leads the immunotherapy and immunology program that was supported by Happy Lungs. And really what I want to do is focus on our T-cell engineering work to try to target these RET fusion positive lung cancers. So one of the simplest demonstrations that the immune system is constantly protecting us from cancer is when you take mice that have an immune system and you compare them to mice that don't. You expose these mice to a chemical carcinogen, and over time what you see is that the mice that don't have an immune system develop cancers much more quickly. But you can see that even the mice that do have an immune system do develop cancer at a delayed onset. Now, what tends to happen is that in the early stages of the cancer, the immune system is quite effective at both seeing and eradicating this cancer. Then you have this equilibrium phase where there's a back and forth between the immune system and the tumor, and then eventually the tumor evolves and the immune system is no longer able to get rid of it. But as you can see, there are still immune cells there. They're just not doing what they should be doing. And here are these three phases overlaid on the tumor growth. So if you actually take a tumor and slice it and look at what's in there in terms of immune cells, this is what you see. Each of these colors represents a different type of immune cell. So you can see there's a heavy infiltration of immune cells here, and we're particularly interested in T cells, and these are the cells here that are shown in red. So once again, significant number of T cells that are found within these tumors, yet these tumors are still developing. So here's a video showing what may be happening. You'll see in green are the tumor cells, and red are the T cells. And what you'll see, despite there being the same number of T cells on the left and the right, is that On the right, the T cells encounter the tumor and they just move on because they're not able to see and act upon those tumor cells. Whereas on the left, when you have the proper T cells present, those T cells are actually able to recognize the tumor, and you can see the tumor cells kind of shrivel up and detach from the plastic. Now, the way this happens is this process called antigen presentation. All the cells in your body, including the tumor cells, are constantly presenting antigens or displaying antigens at their cell surface that reflect the state in which they are. And so a good analogy for this is really, it's kind of like an ID badge. So the immune system is constantly scanning each one of these cells and looking if they have a badge that suggests that they're healthy or they're not an intruder. The immune system moves on. If it's a badge associated with cancer or some sort of infection, then the T cells will then go on to recognize those cells and destroy them specifically while sparing healthy cells. Now, the way this occurs is through the T cell receptor. Here's a schematic showing the structure of the T cell receptor. And there's an interaction between the different regions of the T cell receptor and both the peptide, which is the antigen that we tend to go after, and what's called the MHC molecule, which is basically displaying that antigen at the cell surface. And as a result of this interaction, what happens is that you have the activation of the T cell and the subsequent lysis of the target cell here. Here's another video showing what's happening here. The T cells are in purple. You can see that as they kill the tumor cells become red here and they start to shrivel up and die. And then here's another example here. You can see a T cell doesn't necessarily kill only a single target. Here you have a T cell going and killing multiple targets. So they're extremely effective when the right T cells are actually present. So one way to leverage these T cells is through what's called TIL therapy, and TIL stands for tumor infiltrating lymphocytes. What happens here is that patients have surgical resections to resect their tumors. Those tumors are cut into small pieces. The immune T cells that are present are grown in the lab to extremely high numbers, tens and hundreds of billions, and then they can be reinfused into patients to try to get rid of what's remaining of the cancer and protect from the reoccurrence of the cancer. And there's been a clinical trial in lung cancer that was presented a couple of years ago by Ben Creelan's group at Moffitt. What you can see here, each one of these lines represents a patient, and the percentages are the change in the size of the tumors over time. And what you can see is that some patients had complete clearance of their tumors here in blue, and many patients did respond, but eventually the tumors tended to progress and they even developed new tumors in some cases. So it works, but it doesn't work for everyone. Now, importantly, when you're talking about lung cancer, obviously you're not talking about an isolated tumor. You're talking about a tumor that's found within the lungs, and the lungs are constantly exposed to the outside environment, to infections and carcinogens and so on. And so that matters. And in fact, when you look at the T cells that are infiltrating these tumors, you can see that it's a minority of these T cells that are actually relevant to eradicating tumors. Here's a figure on the left from a paper from a few years ago showing that there's only about 10 to 15% of T cells that infiltrate the tumor that are actually able to see and kill that tumor. And on the right is what we call an ELISpot. And what's happening there is that on the left part of that, you have PBMCs, which are cells from the peripheral blood of a lung cancer patient. And on the right are the TILs, so the tumor infiltrating lymphocytes. When you expose those cells to viral antigens, what you see is that in the TILs, there's a considerable of these T cells that are actually able to see and become activated upon exposure to viral antigens. And these are antigens that are obviously irrelevant to tumor clearance. And so what can we do? Well, we can engineer these cells. So you have constantly these T cells that are circulating throughout your body that recognize influenza, maybe COVID and so on. These cells ignore tumors when they tend to encounter them. But what we can do is that we can engineer them. We can basically carve out their T cell receptor and replace it with the T cell receptor that's targeted specifically to cancer, so that now what you have is a supercharged army of T cells that are both able to see and kill tumors the next time they encounter them. Now, you may have heard about chimeric antigen receptors or CAR T cells. Those have been extremely successful. We're talking about something a little bit different here. This is T cell receptor engineering. And the important distinction here, one of the important distinctions, is that with a CAR T cell, although they're extremely effective, they can only target the antigens that are found at the surface of a cell. And so that's only about a quarter of all the proteins that are found within a tumor cell, whereas a T cell receptor actually has access to the entire proteome. And so it has a much broader range of potential antigens. And so over time, we've developed this workflow to generate these T cells and to engineer them. I'm not going to go into detail here, but I'm happy to answer questions later on if there are any. And importantly, once again, as we talk about these T cell receptors as opposed to chimeric antigen receptors, it's not all to have the right mutation or the right antigen. Here you have an example with 6 different patients with the same antigen. But when you're talking about TCRs, because these antigens need to be displayed on these MHC molecules, you need to actually have this additional step to filter for patients that may be eligible, which is the presence of these HLA molecules here. And you can see in this example, out of the 6 patients, there would be 2 that would be for whom this antigen would be relevant. And so to account for the greatest number of patients possible, we tend to focus on the 10 most prevalent HLA alleles in the United States population. And based on our analyses, at least one of these is found in upwards of 90% of patients in the US. So what's most important is identifying the right antigen. And so there are a couple classes of antigens when you're talking about tumors. On the left, these are called tumor-associated antigens, and these are basically proteins that are found in the tumor at higher levels than they are found in healthy tissues. On the right, you have what are called tumor-specific antigens or neoantigens, and these are antigens that arise from mutations found in the tumor that are actually absent from normal tissues. And we have efforts targeting both of these types of antigens. Now, when you talk specifically about RET fusions, as was discussed previously by John and by Alex, these fusions account for approximately 2.5% of lung cancer, and the patients with these fusions tend to be unresponsive to immunotherapy. Now, as was shown previously, the patients that respond to immunotherapy tend to be those that have this higher tumor mutational burden. And the reason for that is that these mutations tend to make the tumor more visible to the immune system, and they provide something that distinguishes the tumor from the immune system, distinguishes the tumor from the non-tumor, that the immune system can kind of use to distinguish the two and to specifically eradicate tumors. Now, when you look at RET tumors, on the right here, you can see they have a very low tumor mutational burden. And so that may be part of the reason that, once again, where they don't respond to checkpoint immunotherapies. They also have low expression of this PD-L1 protein, which is the target for many of these immunotherapies. So that may be part of the reason why these tumors don't respond very well to classical immunotherapy. Now, thankfully, we have these selective RET inhibitors that are quite effective, selpercatinib, pralsetinib. You can see the responses here, but as we know, they don't work forever, and eventually patients may progress. So what we've been doing here, here's a snapshot of the different RET fusions. As was highlighted by John, these are extremely heterogeneous and diverse. There are many different fusion partners, and there are many different breakpoints. And so what we've been doing is to try to identify antigens that arise both from these breakpoints, but also from the regions that are consistent across each of these RET fusions. And we're using those to try to stimulate the immune response. So here's an example of our data here. This is using what we call an epitope. It's basically a part of the protein that we consider to be immunogenic or to be able to trigger an immune response. If you've never looked at these plots before, this is from flow cytometry. The x-axis shows the cells that are dying, and the y-axis shows the cells that have been dead for a while. So basically, these are all tumor cells, and as they die, they tend to move to the right here and then move upwards. And so what you can see is that when the cells have this antigen, the T cells are actually able to kill them quite effectively. And importantly, we're actually able to find T cells that are able to kill not just the cells that are pulsed with this antigen, which is what we use as a positive control, but these are tumors that express endogenously, in this case, the K24R11 fusion presented on HLA-A2. And here on the right is the quantification. You can see the ability of these cells to specifically eradicate the tumor cells that harbor the fusion because of the upregulation or the higher expression of RET within these tumor cells. Now, we've also done this with HLA-A2402. This is another highly prevalent HLA allele in the US. It accounts for about 17% of patients. And here, once again, what you can see is that our T cells are able to kill tumor cells quite effectively when they're exposed to the tumor cells that express this fusion. Now, another antigen we've been looking at that appears to be relevant here is this antigen called FOXM1. And what this plot shows here is the level of FOXM1 in many different normal tissues on the left in the different colors here, and in different tumor types here in gray on the right. And what should be seen from here is that the brain-associated tissues and all the critical tissues have extremely low expression of FOXM1 here on the left, whereas all the others are a little bit high, are much higher, as are the different tumor types. Now, in the context of RET fusions, we've done some analysis looking at FOXM1 specifically. What you can see is that tumors that don't harbor RET fusions, first of all, they have higher expression of FOXM1 than does the adjacent normal tissue. And then the tumors that have RET, consistently, the expression of FOXM1 is higher within these tumors. But not only that, FOXM1 actually appears to be upregulated in resistance to selpercatinib and pralsetinib, as can be seen in this Western blot here. And you can see that the parental cell lines don't have any FOXM1, but when they become resistant to these two selective RET inhibitors, they start to present FOXM1 at the cell surface. And so far, we've had some success generating T cells that recognize these antigens. On the left is one of our TCRs that recognizes FOXM1 on HLA-A2. HLA-A2 is found in 42% of the United States population. On the right, for A2402, again, 17% of the US population. You can see that in both these cases, we have T cell receptors that are quite sensitive and specific. And so here's where we stand currently. We've been able to do this successfully for many different targets, including EGFR, KRAS, HER2, and so on. In the context of RET, we've been making some good progress here in developing our RET-specific TCRs, as well as these TCRs targeting FOXM1. And we hope to move these forward in coming months as well. And here's an overview of the patients for whom these treatments would be compatible, again, focusing on EGFR, HER2, KRAS, and FOXM1, which would be relevant in the context of RET fusions. When we crunch the numbers, what we see is that our TCR library would be compatible with upwards of 150,000 lung cancer patients in the US per year. And that's all I have. I want to acknowledge the members of the lab who generated all of this data. And once again, thank Happy Lungs for the support.
Thank you so much, Dr. Ruben. This is great. And I think we have some questions on the chat now. I don't know if Dr. Heimak is here, but if not, we can send this question to. Okay.
Yeah.
Oh,
Yep,
perfect.
I'm here. I'm here, Renee.
Hi.
With the question.
Hi, John. Okay. Yeah. Stephen asked, Dr. Heimak, thank you. What you have said about K15 and R12 is also applicable to K16 and R12.
We assume it is, yes. We've the secondary mutation. So we've seen basically the same sensitivity for 1512 and 1612 across the board, with some minor exceptions. For the resistance mutations, we put those all into 1512. But from what we know about the structure, we expect everything about the resistance mutations would be exactly the same for 1612.
Great.
Great. I can take the next question. So the next question is there any enthusiasm for using combination therapy early to prolong targeted therapy activity? And I think the conceptual answer is yes. If we identify a mechanism of resistance that's independent of RET, that emerges with a good frequency on RET inhibitor monotherapy, then we could do this, but we often run into issues of side effects. And so when you combine two treatments, you can have more side effects, and that could impact how long patients stay on a therapy because it's not tolerated as well. But if we were to find two treatments that we could combine without bad side effects, and there's good data to support it, then this is something that we would be excited about.
Great. Thank you so much. There is another question. Is there any research to find immunogenic surface antigens and then making an mRNA approach to attack these in RET non-small cell lung cancer?
So the project that was funded by Happy Lungs does support the identification of surface antigens. That's work that Justin Gaynor and myself are performing as we speak and have been focusing on. By doing this, we're able to identify antigens, both surface antigens that would be targetable with chimeric antigen receptors, or MHC-presented antigens that would be targetable with TCRs or with vaccine approaches. And so we're not so much focused on the actual vaccine approach as of now. We're really at the stage where we're identifying relevant antigens. And once we have those antigens, we're able to target them in different approaches, including mRNA. So that'll be a subsequent step.
Great. Thanks so much, Dr. Ruben. And there's another one. How common it is to lose the RET mutation after targeted therapy and progression?
So to lose the resistance RET mutation or the initial RET fusion. If it's, this has been asked several ways, but one question is if the original RET fusion or mutation that's driving the cancer can disappear at some point in time. I think that a lot of what we know from many lung cancers with drivers is that often the original fusion or mutation will stick around, but there is some data to show that perhaps in some cases you can lose it. It's a very small number of observations. And some of these might be within the same patient. So we had a patient where we were looking at liquid biopsies over time, where it seemed like there was a clone that became much more prevalent on the liquid biopsy that appeared to be RET wild type, meaning it did not have the original fusion, but it had KRAS, while the original RET fusion containing clone appeared to be suppressed. For RET mutations, resistance mutations, I assume that these can pop in and out depending on what treatment the cancer has gotten, right? Because sometimes these clones can reorganize. So I think there's a little bit of variability there depending on what the patient last received.
Yeah, and I'll just add a couple comments, and I agree with everything Alex said. In addition, sometimes the tumor doesn't lose RET fusions, but it changes what we call its lineage. It shifts to another type of histology, like towards small cell or towards squamous or towards EMT. And when that happens, it doesn't care about RET anymore. It becomes dependent on other things. And that's where we need an entirely different treatment strategy. You could essentially forget about RET, and that's where you need to, target what's on the cell surface or some other approach. So we call that losing RET dependence, and that does happen as well.
Rena, you're muted.
I'm sorry. There is another question. What is the state of research on drug-tolerant persistent cells and targeting them in some way?
Yeah, I can talk a little bit about this. So we've just started taking a look at RET drug-tolerant persisters, but we've also got some experience from looking at drug-tolerant persisters in EGFR as well. So Monique has led a lot of these efforts, and we see that drug-tolerant persisters often turn up unique proteins on the cell surface. Previously, we saw for EGFR, CD70 was turned on, and that you could target CD70 with either an antibody drug conjugate or a CAR T or an NK CAR. Well, now we see for RET fusions, it looks like CD70 is also turned on. So for that reason, we're thinking about ways we might combine a CD70 targeting approach with the TKI. The idea being the TKI would eliminate 99%, and hopefully the CD70 approach would work on the rest. Monique, did you want to add any additional comments there?
No, I think that summed it up really well.
Perfect. Thank you all. I think there's no more questions for now, but if you have another question, we are happy to email your questions to the investigators and get back to you soon.
Right. You can send your questions to info@happylungs, and we can that's also on the website if you just click on it, and we can forward questions or answer them. Thank you to all the investigators, the multi-centers who are doing this amazing work, and to all of you who have given your time today to explain it to us, the patients and the donors and the people that are really relying on your work to continue living with cancer and doing well. We hope everybody has really happy, healthy holidays ahead and stays well. So thank you to everybody, and we will see you in the new year probably.
Thank you so much.
Thank you, everyone.
Happy holidays, everyone.
Happy holidays. Thank you
Take
all.
care. Thank you.
Bye-bye.
Thank you.